Okay, so we'll wait for the last stragglers to go out. And the students who haven't gone back to run back to the lab, as Eric suggested. Okay, so this was amazing. Thank you so much for those really brilliant lectures. So varied. I think I'd like to start with you, Professor Botstein. Yes. I'm going to go a little wider in my questions as opposed to specific details, though we may still have a lingering question or two for Professor Lander. So if that comes up and when we open to the audience, we can certainly entertain those. So you showed a little bit of history. You showed Monod. And if you were giving advice to the students here in the audience, those of us who have been in the field a little longer, what do you think is the single most important discovery that enabled the Human Genome Project to be tackled? Or, in other words, what was the turning point?
Oh, for sure. It's Sturtevant 1913. Yeah. Okay? Sturtevant 1913. Does Sturtevant mean anything to you? Does the microphone worketh or worketh not? Not. I will Alfred Sturtevant. It works. It works? It works? Alfred Sturtevant wrote a paper in 1913. He was a sophomore at Columbia University. He happened to work in Morgan's laboratory in the famous fly room. And one night he took home all the data that had been collected about a cross which involved 3 different morphological markers that he could tell apart in the Drosophila. And they had counted very carefully thousands of progeny. And he went home and he realized that the frequency of recombination could be put on a linear scale on a map. Such that if the frequency of recombination was less, then the marker, the 2 genes were put closer on the map, and if it was more, they went further on the map. Okay, and he showed that, and then this is the really brilliant part. He showed that the double crossovers were roughly the product of the single crossovers, that you could tell the in-between marker by being the minimum recombination class. And when we first proposed mapping the human genome, we said nothing different from that. And it's still the way it's done. And the human genome, in fact, would not have been sequenced had we not had a useful map, because most of the physical maps were very iffy, especially long range. So I would say that without question, it was Sturtevant 1913, the undergraduate homework.
Which is taught in my undergraduate biology course, because it is, I agree with Botstein, that is, the seminal notion that you could find out where something was in the genome before you knew what it was.
It's the concept of genetic linkage. It's if there's a fundamental theorem of biology beyond Mendel, that's it. Fantastic. Do they teach you that in Israel? Good.
They were just shy.
There's a more remarkable thing about Sturtevant, even than that he was able to do that, although that was really one of the stellar achievements. There are two interesting things about it. First of all, he never got a Nobel Prize, which I always thought was a terrible travesty. He lived well into the 60s and never got it. That was one thing. The other thing was that he did all this with these Drosophila eye colors. How many of you have seen the Drosophila eye colors? Mm-hmm. They're hard, right? He was colorblind. He was a straightforward deuteranope, and somehow he knew how to. He could distinguish vermilion from wild type.
Craig, the next one is for you. So you talked about several new frontiers, and clearly you painted a picture in many ways of the future for us, perhaps, in algae. But as many students who are here in the audience who are trying to be practical, they're trying to strategize what field they should pursue other than perhaps learning Mandarin, which is the new hot thing to do. Where would you advise your students to go in the future?
So, in fact, that question came up in some of my earlier discussions with students, and my answer was become an expert in something. And it really doesn't matter when you look at, I think, the Human Genome Project as a whole, or what the Celera team did. It was an integrated team of computer scientists, mathematicians, molecular biologists, biochemists, technologists, a few economists, even a geneticist or two. The key was people of different clear-cut expertise bringing their expertise to the table to work in an integrated fashion. And what I indicated was, for our team, everybody on the team knew that if they failed at their discipline, if the algorithms didn't work, if the sequencers didn't work, if the automation didn't work, it didn't matter what part people were doing. They knew if their part failed, the whole thing failed. And I think that's true for the future view of science. But if you're not an expert at something and you don't have a thing to contribute, you're just in the way.
Okay, so students don't get in the way. So Eric, I have one for you. You talked about the epigenome, and everything is looking much more complicated than we ever envisioned that it would be. There's no question what we discovered in the last 10 years is in many ways much more than what we knew before. So when you're thinking about where drug design is going, curing our diseases, do you build a mega drug that's going to attack, looking at the link RNAs, looking at expression, looking at single gene defects? How do you see disease being combated when it's so complicated?
So, look, the first thing to say about all this complication is science progresses by being overly simplistic. I mean, is it really more complicated than we thought? Did anybody really think that having the genes laid out, suddenly all the rest of it would be easy? No, we just temporarily delude ourselves that this next step is so damned important that we're all going to work our tails off to do that step, because without that, you're not going to get it done. There'll be more complications beyond this and more complications beyond this, but these sets of complications now seem more tractable. And I suspect the human organism, it took 3.5 billion years to get a human. It's not at all unreasonable that it could take more than a decade to kind of understand it. So I'm not so worried about that. What I think, I'm sort of a very practical person. What do we have to do to make drug development a little bit better, and then a little bit beyond that? I think what we have to do is bring the same spirit as we brought to the Human Genome Project of making things efficient, collecting data and integrating it in certain ways. So, for example, the ability to take cancer. I'll give you a project that a bunch of people are working on. 1,000 cancer cell lines. Get the complete genome of 1,000 cancer cell lines. Get the complete dependency on genes across each of those 1,000 cell lines. Knock out each of the 20,000 genes in each of the 1,000 cancer cell lines. That's doable with parallel RNAi. It's not so hard to do anymore. Get the complete epigenomic pattern. Take all known FDA-approved drugs and everything in trials and run them against those 1,000 cell lines. That's all not that hard to do. It is all being done, and it'll get done in the next year or two. Then you can begin to ask, all right, these drugs specifically kill these cancer cell lines. What's so special about those cancer cell lines in terms of their particular mutations? And it's beginning to work, and it's beginning to point us to things. And I think if we imagine that this is a project that is going to take, it will take a couple of decades, but a couple of decades to be able to tease apart a set of combination therapies for cancer, in retrospect, that's going to be the blink of an eye. So I think over the next 5 years or so, the full set of dependencies will get known, and we'll know what the key Achilles heels are of different types of cancer cells as a function of their genotype. We'll know a bunch about drugs. We'll then start to ask about combinations. There are ways to bring genomics to bear to find resistance. You can put in extra copies of every gene in all of these cell lines, and the thing is, the young people are thinking of those things now. They're thinking about bringing that to the chemistry that's involved. I think what genomics really taught us was that systematic attacks on things, comprehensive data, can be very powerful. And even then, we have to be very careful not to promise that we're going to go cure all cancer. We're not going to go cure all cancer anytime soon. If we get 15% off the death rate of cancer, we should be very proud of that, and they'll get more off that. And that's how real progress is made. Sometimes, the Journal of the New York Times likes to write articles saying that, what, we sequenced the human genome 10 years ago and there's still disease? I mean, they do. Trust me, they write this article. I think this is a great intellectual program that's moving forward faster than I could have ever imagined. And that still might not be as fast as somebody who has cancer today might like. But when you think about the 75 years it took from the discovery of the germ theory of disease to broad antibiotics cheaply available, wasn't bad use of 75 years. I think we'll see a similar thing here.
Professor Buxton, you wanted to add a comment.
I agree with all that, but I don't completely agree with the enthusiasm for how much is going to get cured. I think of this not as blitzkrieg, but a war of attrition. I'm a microbiologist originally, and even the microbes, you understand the microbes are winning in some areas. Some of the things that Craig is finding in the hospital are really not good for you, and we don't have any antibiotics left against them. And I think we should be paying attention to the fact that evolution is about survival of the fittest. And the staphylococci have their methods, and we have to have a. That is not to say
microbes are a much different problem. We will not win against microbes because microbes keep sharing their learning each further generation. Every cancer starts naive. It doesn't know what we're going to have. So in the long run, cancer turns out to be an easier problem. Microbes are really smarter because they really keep being selected.
I'll sign up for that. What you say is right. Nevertheless, the cancer is not so simple.
It's not easy, but at least we win in the long run because it doesn't know what we've got, whereas microbes do know what we've got.
Somebody asked, what is the substructure of tumors beyond the major classes? And, I don't know yet, and nobody knows, but it may be very diverse. And the more diverse it is, the more you'll have to get private drugs for private tumors. And it's going to be, think trench warfare, don't think blitzkrieg.
So let me add to the complexity issue, because I'm actually, I'm much more optimistic the more complexity we discover, because I started my science career during the 1970s, which I call the cyclic nucleotide era, where everything in human physiology and plant physiology and any physiology you can imagine was explained by two chemicals going up or down in cells.
So, yeah.
And so Eric talked about some simplistic models. I don't think it could get much more simplistic than that kind of thinking. And there were tens of thousands of papers published in the scientific literature about it. The more we understand the components, this nice dissection that Eric showed of the human genome, having the microbiome, knowing the chemical diversity, all that gives me optimism. The more components we know, the more our chances of doing something meaningful increase, especially if we can mathematically approach the complexity. The only thing that gives me pause, as long as we're talking about cancer, is Bert Vogelstein's recent data showing the cell-to-cell variation looking at the cancer genome makes it extremely unpredictive. If that gets replicated, that could be the most depressing study to come out in a long time.
It's back and forth. But we are definitely better off. Nobody should want to live from the point of view of the prospects of what happens to you when you get sick 50 years ago, 100 years ago, let alone 500 years ago.
Yeah, and let's also, to Craig's point about complexity, think about HIV, right? there wasn't so long ago that we were deeply, deeply depressed about ever dealing with HIV because it mutated so fast. It had vastly more diversity, has vastly more diversity than any of these microbes and any of these cancers. Any one drug, the chance of mutating around it was, I don't know, one virus in 10 to the 6th or 7th could mutate around it. And there were 10 to the 15th viruses there. Any two drugs, you multiply 10 to the 6th times 10 to the 6th, and you still got, not as much as the 10 to the 15th viruses. You get 3 drugs, you win. So there is something to be said for the power of combinatorial attack used simultaneously. But that's only going to come slowly. We're going to get a drug here, and there's going to be resistance to it. You've got a year out of that drug, and then people will figure out how to combine it with something else and get the power of multiplying the numbers, and onward like that. I'm with David that it is trench warfare. It's going to take decades. There was somebody who said kind of famously in 2000 and whatever that he was going to eliminate death and suffering from cancer by the year 2015.
Yeah, right.
And this was nuts.
Soon.
This was just nuts. But the idea that we're going to make this incremental progress and it adds up to something, it's right.
Well, one of the main challenges that we may face is not even the practical issues of science, but more how do we distribute all these drugs? How do we make it available? Even deep sequencing today, if we're going to be able in 5-10 years be able to work on some of our diseases because of our sequence, is that going to be accessible to everyone? So that's going to be a challenge as well, not just the discovery of science.
Yeah.
It's in our hands.
$100,000 a year per drug per patient is not going to be a viable model.
No, and the people are sort of only half-heartedly working on malaria drugs in large part because they don't think that the people who need them can afford them. And so that level of problem we didn't even start with. It's not our expertise. But you can't just put your head in the sand. This is a problem.
I'd like to take the opportunity to ask: is there anyone who has a question for Eric, for Professor Lander?
Or anybody? No.
The last comment that you made about the 3 different drugs that you compare to cancer, aren't you going to face the problematic of mixing science and the regulatory, the way that it is set right now, the FDA to do phase 2 trial of such drugs?
So the question, for those who might not have heard it, was, all right, fine, the science sounds good, but what about the regulatory environment? What regulatory environment is going to make it possible to test all these combinations, et cetera? I remind us, what regulatory environment let us test the AIDS drugs? Well, the original regulatory environment was not very favorable for testing AIDS drugs, and AIDS patients acted up. And the whole concept of the accelerated approval pathway of the FDA came from the AIDS activists who said, maybe we should be willing to approve a drug based on a surrogate indicator rather than waiting for proof of long-term survival before the drug could be approved. They changed the regulatory pathway. So I was at a meeting 2 or 3 days ago where there was a couple of cancer people, and we were talking about the fact that mutations are being found now in patients. And the mutations, with a reasonable probability, some non-trivial percentage of those patients, there's a drug in clinical trials against the protein that's mutated. It'd be very nice to try that drug on that patient, but it's in clinical trials. What are you going to do? How do you get that drug to that patient? They're not on the right protocol. Well, the FDA has a single patient IND pathway where you can get approval for a single patient. And you can do that in 48 hours. But then you say, the pharmaceutical companies might say, I don't want you screwing around with my clinical trial by generating data that are going to mess up my approval for some patient who might die because they're late. But then you say, couldn't the FDA agree that data collected under the single patient IND might be in a very different category with regard to registration of a drug? And now some folks at the FDA are saying, we could do that. That's not crazy. So it turns out that regulatory pathways are not written in stone. If there is enough of a groundswell of scientific and patient interest coming together to say, We need to be able to test new drugs, new combinations, collect those data, share those data, and the following three things stand in the way, we will somehow change them because it matters enough.
Yeah, I agree with that. My favorite example is growth hormone. In the spirit of disclosure, I used to work at Genentech. When growth hormone was before the FDA, it was only the second recombinant drug, and they were very, very reluctant. And they dragged their feet, and they dragged their feet, and they dragged their feet, and they dragged their feet some more. And it was really very depressing. But then the kids who were getting growth hormone were getting it from ground up cadavers and was purified as a protein. And then somebody noticed that some of these kids got Jakob-Creutzfeldt disease. Okay, after the second case report of Jakob-Creutzfeldt disease, we got a call from the FDA. We said, we've decided maybe it's a good idea. We're going to remove the cadaver material from use, and maybe you guys can make enough. Of course, we had plenty, okay? And that was...
Yeah.
So it's partly science and it's partly politics. But one should not underestimate the importance of the science. This is not a standard commercial regulatory thing. It really is about the science. And so these agencies are very important because if it weren't for these kinds of regulations, then it would be all about marketing. As it is now, it's only partly about marketing.
Craig, do you have anything you'd like to add in closing remarks?
To that discussion, I can't help.
When's the next time you're going sailing?
Well, we're getting ready to go down to the Amazon basin, hopefully. What I've argued many times is Darwin couldn't have possibly done what he did in today's environment, where you have to have these massive permits. We have to negotiate through the State Department with each country for anything within 200 miles of the coast. And when you look at the Pacific Ocean, for example, with every island, there's a lot of the ocean that's owned. When the expedition went from the Galapagos across the Pacific, as we sailed into the Marquesas, we were immediately arrested by the French police because they had a political debate with the Tahiti government who controlled the water there. But the notion is there's roughly a 1-knot current moving across the ocean. So as soon as that water moves into the 200-mile boundary, it goes from public organisms to French genetic heritage. And these are real challenges.
But it loses nationality a little ways down.
Yes. Well, it's permanently altered once it was French. But,
Well, thank you. I think what the clear message is that we really need to integrate as many disciplines as possible, and certainly something we're trying to do here, I think successfully at the university, and your contribution in these 2 days has really been tremendous, so I want to thank you. In closing, I'll just say that another tremendous advantage of the Dan David Prize is that the prize winners do have to give up part of their winnings to scholarships. So at 5:30 there will be a scholarship award ceremony for those of you who would like to join. And again, thank you so much to the three of you. It's been incredible.